Hello, I'm Les Recio, the CSO of ScitoVation. Today I'll be talking about HepaRG cells as a new approach methodology for 21st century animal-free genetic toxicology testing. I'll be illustrating this using a follow-up to a positive response that we observed with human TK6 cells using the compound naphthalene. The publication that just came out is shown here, genotoxicity assessment in HepaRG cells. Or the short title would be rat liver S9 P450 bioactivation without phase 2 conjugation can cause genetic toxicology testing false positives, and we believe this is the case with naphthalene. We examined naphthalene cytotoxicity in TK6 cells with S9 compared to what we observed in primary human hepatocytes, and that data is shown here. This was done more than a decade ago, where we used flow cytometry-based micronucleus assay and demonstrated that naphthalene is genotoxic with S9, inducing a positive response in the micronucleus assay at concentrations of 5 micromolar and greater. It was also cytotoxic, inducing a fairly significant cytotoxicity at 30 micromolar, corresponding to about 50% survival. Simultaneously or in parallel, we were also examining naphthalene and cytotoxicity in primary human hepatocytes, and that data is shown here, where the first thing you can tell right off the bat, where we examined both ATP, LDH, or glutathione depletion, that there was a dose scale difference of about tenfold. So with S9, there's about a tenfold difference in the cytotoxicity we observed as compared to what we observed in primary human hepatocytes based on micromolar concentration, and the two publications are shown on the bottom. Well, we believe that phenobarbital beta-naphthalene-induced rat liver homogenate causes bioactivation without detoxification, and this can cause a false positive. Phenobarbital and beta-naphthalene rat liver S9 is used for biotransformation for in vitro hazard identification genetic toxicology bioassays. This has phase 1 metabolism only, and it's not relevant to humans. It's a rat liver induced that is not comparable to what you might see in a human hepatocyte, for instance. Phase 1 metabolism tend to be oxidation reactions that bioactivate compounds to reactive intermediates. They can create or form functional groups that are substrates for phase 2 enzymes. And phase 2 enzymes conjugate, increase ribosylate, detoxify, and more importantly, they make xenobiotic available for intracellular transporters. So in vitro with rat liver S9, you have phase 1 and phase 2 metabolism. However, in vivo or in vitro metabolites, you have both phase 1 and phase 2 metabolism. They are linked biochemically, and also phase 2 metabolites are substrates for elimination and excretion by transporter enzymes. This is shown here at the bottom, where basically you have naphthol and naphthylquinone. Naphthylquinone can form reactive intermediates. However, they are glucuronidated or conjugated with glutathione in vitro, and we were able to show this in vitro using human hepatocytes, where in rats, the glutathione conjugates of naphthylquinone are the primary metabolites formed. The regulatory genetic toxicology test battery is shown here. In vitro tests that are devoid of metabolizing enzymes, so that is accomplished by adding rat liver S9. However, there's no phase 2 conjugation. It's P450 bioactivation using NADPH as a cofactor. These assays were intended as hazard identification, not really quantitative risk assessment assays. We do have in vivo testing as follow-ups to these in vitro tests. However, there's no intermediate. There's no metabolically competent intermediate that can be used. You basically go from in vitro positive response and you jump into a positive, examining a positive follow-up using an in vivo test, either the micronucleus comet or more recently mutagenicity testing using transgenic mouse or error-corrected DNA sequencing. There is no intermediate assay. Therefore, we believe that metabolic competent HepaRG cells can serve as a genetic toxicology test system because I believe there's a need to develop human-based in vitro bioassays that capture xenobiotic biotransformation, both phase 1 and phase 2 metabolism, and enzymatic induction to replace, refine, and reduce complete reliance on animals as a sole follow-up to in vitro genetic toxicology test battery positive responses. So the only alternative we have right now when you have a positive response is to go into an animal, and many industries don't want to go into animals and need alternatives. HepaRG cells have both phase 1 and phase 2 biotransformations, as I commented, drug transporters. They also express nuclear receptors, which are part of the non-genotoxic modes of action. Their cellular compartmentalization becomes very important for reactive oxygen, and they can be grown as spheroids. And this just to illustrate the point that TK6 cells shown here basically look like a bicycle tire, small nucleus, very little cytoplasm, as compared to hepatocytes, which you have a very high cytoplasm to nuclear ratio, and that cytoplasm is chock full of endoplasmic reticulum and cytosolic enzymes that are involved in drug metabolism. We have used HepaRG cells, and myself, and with a collaboration with Bevan Engelward and Carol Yock, qualifying a number of endpoints that can be measured in vivo, can now be measured in vitro using HepaRG cells as a replacement for animal toxicology, genetic toxicology. So we developed a kind of a standardized protocol where basically we use the thawed no-spin HepaRG cells, incubate them in a 96-well collagen-coated plate for 7 days, let them express high levels of P450, and then we treat them on a 3-day repeat dosing schedule and measure a number of endpoints at the end. Right, what I'll be talking about today are the Comet chip assay, and with epidermal growth factor, we are able to get the HepaRG cells to go into cell division and measure a micronucleus. So 3 days of exposure, right after the last exposure, 6 hours after the last exposure, we do the Comet chip assay, and then give the cells an epidermal growth factor and measure the micronucleus 3 days afterwards. First study shown here is using the Comet chip. It was developed at MIT by Bevan Engelward, and as you can tell, there's absolutely no increase in the induction of DNA damage as measured by the Comet assay, percent tail DNA, completely flat. In HepaRG cells, no response whatsoever. Even if we cause a cytotoxic dose here, killing about 50% of the cells, there's still no increase in genotoxicity in HepaRG cells exposed to millimolar concentrations up to 1 millimolar of naphthalene. So with the Comet assay, DNA damage assessment, there's absolutely no indication of genotoxicity by naphthalene. We then turn to the micronucleus assay using a flow cytometry-based micronucleus assay. Again, that data is shown here. Absolutely no increase in the micronucleus induction in HepaRG cells using a flow cytometry-based micronucleus assay, even as we get it to cytotoxic concentrations right around here where the cutoff is, we get absolutely no increase in micronuclei. We do get somewhat of an increase as we exceed the cytotoxicity down to 20%, which fits into the hypothesis that these kinds of compounds at high concentrations can induce genotoxicity, but it's irrelevant, highly cytotoxic concentrations that aren't used in regulatory genetic toxicology testing. So sum up, rat liver S9 shown on the top here, as we metabolize, as naphthalene gets metabolized by P450, it generates a number of cytotoxic and genotoxic metabolites, primarily naphthalene epoxide. However, that can cause covalent binding, cytotoxicity, and the naphthoquinones that from naphthalene, which can cause cytotoxicity, DNA damage, or chromosome aberrations due to reactive oxygen. This is what happens with S9. However, in a metabolic competent cell or in vivo, where you have both phase 1 and phase 2 enzymes, these toxic metabolites are readily detoxified by phase 2 enzymes, either glutathione conjugates. Naphthol can be conjugated with either sulfate or glucuronide, preventing the formation of naphthoquinones. And if naphthoquinones are formed, they can be conjugated with glutathione. So there is a number of detoxification steps that are not present in rat liver S9, which causes a cytotoxic and genotoxic response in the micronucleus assay with TK6 cells. So in TK6 cells, naphthalene is genotoxic without phase 2 conjugation. However, when you have a metabolic competent cell that uses both, has phase 1 and phase 2 enzyme, it's non-genotoxic. We did a benchmark dose with rat liver S9, but it's clear that it's not relevant to what we observe in human hepatocytes or in HepaRG cells. So, and it just and also this corresponds to what we see in vivo, where naphthalene is not genotoxic in vivo in the liver or in the bone marrow micronucleus assay. ScitoVation, the 21st Genetic Toxicology Program, is focused on using computational tools, the micronucleus assay and genomics with in vitro to in vivo extrapolation modeling to avoid a point of departure. The micronucleus assay in HepaRG cells and TK6 cells are a primary bioassay that we have been focusing on at ScitoVation over the last year. And finally, to sum up, ScitoVation is focusing on integrating new approach methodologies into 21st century toxicology, as well as genetic toxicology. We believe that metabolic competent HepaRG cells are a useful follow-up to in vitro positive responses that can be used in quantitative risk assessments, as we do here at ScitoVation using in vitro to in vivo extrapolation modeling. So thank you very much for your time, and we appreciate your attention today. Thank you.